For a long time, life seemed easy to divide. There were plants, animals, fungi, and all the tiny single-celled organisms people casually called bacteria. But hidden inside that simple picture was a whole domain of life that scientists had been overlooking. Archaea are microscopic organisms that often look bacterial under a microscope, yet at the molecular level they are something profoundly different. They are not just strange bacteria with an old-fashioned name. They represent one of the major branches of life on Earth. Their discovery forced biologists to redraw the tree of life and rethink what it means to be ancient, simple, or advanced. The word archaea comes from an ancient Greek root meaning ancient, and early scientists associated them with primitive life. At first, they were called archaebacteria because they were small, single-celled, and lacked a nucleus, just like bacteria. But that label turned out to be misleading. When researchers compared genetic sequences, especially the machinery cells used to build proteins, they found that archaea were not merely a bacterial subgroup. They formed a separate lineage. In some crucial ways, archaea are actually more closely related to eukaryotes, the group that includes animals, plants, fungi, and humans, than they are to bacteria. To understand why archaea matter, it helps to look at the three-domain model of life: bacteria, archaea, and eukaryota. Bacteria and archaea are both prokaryotic in the traditional sense, meaning their cells do not contain a nucleus. But that similarity can be deceptive. Two organisms can look alike because they are small and simple in structure, while still being deeply different in chemistry and ancestry. Archaea show that outward appearance is not enough to classify life. Their genes, enzymes, membranes, and metabolic strategies reveal a distinct biological identity that was invisible before molecular biology made it possible to compare life at the genetic level. One of the clearest differences between archaea and bacteria is the cell membrane. Every cell needs a boundary, a protective layer that separates inside from outside. In bacteria and eukaryotes, membranes are built mainly with ester-linked lipids. Archaea use ether-linked lipids instead. That may sound like a minor chemical detail, but it changes how the membrane behaves. Archaeal membranes can be unusually stable, which helps explain why many archaea survive in extreme conditions such as intense heat, high salt, or chemically harsh environments. Their membranes are not just packaging. They are one reason these organisms can live where many other cells would fall apart. The first archaea scientists noticed were extremophiles, organisms thriving in places that seemed almost hostile to life. Some lived in boiling hot springs. Others were found in hypersalty lakes where ordinary cells would lose water and collapse. This gave archaea a reputation as exotic survival machines, specialists of the extreme. But that early image was incomplete. As molecular detection improved, scientists began finding archaeal DNA almost everywhere, in oceans, soils, wetlands, sediments, animal bodies, and human microbiomes. Archaea were not rare oddities at the edges of habitability. They were widespread, abundant, and quietly active across the planet. In the oceans, archaea may be among the most abundant organisms on Earth. They are especially important in planktonic communities, where countless microscopic cells drift through the water and drive global chemical cycles. Many archaea participate in carbon fixation, nitrogen cycling, and the turnover of organic compounds. These processes may sound invisible, but they influence the atmosphere, the fertility of ecosystems, and the flow of energy through food webs. When people think about marine life, they imagine fish, whales, or coral reefs. But at the base of the ocean's living systems are microbes, including vast numbers of archaea performing essential chemical work. Archaea are also metabolically remarkable. Eukaryotes mostly rely on a narrower set of energy strategies, but archaea can use an unusually wide range of energy sources. Some process organic compounds such as sugars. Others use ammonia, metal ions, or hydrogen gas. Some salt-loving archaea can use sunlight as an energy source, though not in the same way plants or cyanobacteria perform oxygen-producing photosynthesis. This flexibility makes archaea ecological problem solvers. They occupy chemical niches that other organisms cannot use efficiently, and by doing so they help connect different parts of Earth's biological and geochemical systems. One of the most important archaeal metabolisms is methanogenesis, the production of methane. This process is not known from bacteria or eukaryotes. It is a distinctive archaeal specialty. Methanogenic archaea live in oxygen-poor environments such as marshes, sediments, sewage systems, and digestive tracts. In the guts of humans and ruminant animals, they help process byproducts from digestion by interacting with other microbes. In cattle and sheep, methanogens are part of the microbial community that helps break down plant material. The methane they produce also connects them to climate, agriculture, waste treatment, and renewable energy systems. Archaea are part of the microbiota of many organisms, including humans. They have been found in the gut, the mouth, and on the skin. Unlike many familiar microbes, archaea are not mainly known as pathogens. In fact, as of 2024, only one non-eukaryotic archaeal species has been identified as parasitic. Many archaea appear to be mutualists or commensals, meaning they either help their host or live alongside it without causing obvious harm. This makes them an important but often overlooked part of microbiome science. Understanding archaea may improve our picture of digestion, microbial balance, and how communities of microbes support larger organisms. Archaea reproduce without sex in the way animals or plants understand it. They divide by binary fission, fragment into pieces, or reproduce by budding. Unlike bacteria, no known archaeal species forms endospores, the tough dormant structures that help some bacteria survive extreme stress. Yet archaea still manage to persist across an enormous range of environments. Their survival strategy is not simply to shut down inside a protective spore. Instead, their cellular chemistry, membranes, enzymes, and ecological relationships allow them to remain active or resilient in conditions that challenge ordinary assumptions about life. Classifying archaea is difficult because most known archaea have never been grown in a laboratory. Scientists often identify them from environmental samples by reading genetic sequences directly from soil, seawater, sediments, or biological material. This means much of archaeal diversity is known through molecular fingerprints rather than through fully observed organisms in culture. The result is a strange scientific situation. Archaea may be everywhere and extremely important, yet much of their biology remains mysterious. Each new sequencing project can reveal branches of life that were previously unknown, expanding the map of microbial evolution. The evolutionary importance of archaea goes beyond their own domain. Modern research indicates that eukaryotes evolved from within an archaeal lineage, which means the cells that eventually gave rise to animals, plants, fungi, and humans have deep archaeal roots. This makes the traditional separation between archaea and eukaryotes more complicated than a simple three-way split. In everyday language, archaea usually refers to the prokaryotic members of the group. But in evolutionary terms, they are tied to the origin of complex cells. Studying archaea is therefore not just about obscure microbes. It is also about understanding where we came from. Archaea also matter in practical technology. Methanogens are used in biogas production and sewage treatment because they can convert waste materials into methane-rich gas. Enzymes from extremophile archaea are valuable in biotechnology because they can function under high temperatures or in harsh chemical conditions that would destroy ordinary enzymes. This makes archaea useful not only for understanding life's history, but also for solving modern problems. They help manage waste, generate energy, and provide biological tools for industry and research. The more scientists learn about them, the more useful their unusual chemistry may become. Archaea changed biology because they revealed that the living world is deeper than it looks. They can resemble bacteria in size and shape, yet differ in their membranes, genes, metabolism, and evolutionary meaning. They live in boiling springs and salt lakes, but also in oceans, soil, sewage, animal guts, and human bodies. They produce methane, cycle nutrients, support microbial communities, and may hold clues to the origin of complex life. Archaea are not just ancient survivors. They are active engineers of Earth's systems, hidden in plain sight, quietly proving that even the smallest life forms can rewrite the biggest scientific stories. If you found this helpful, please like this video and subscribe for more explainers.